In this section, I'm going to talk about the tools of a microbiologist. So the first few slides of this PowerPoint show are just pictures, and I want to talk about what each one of these pictures is. This first picture is a loop. A loop, also known as an inoculating loop, is a tool that's used to move bacteria from one place to the next. So typically we would use a loop to culture microorganisms or to transfer microorganisms from one medium to the other medium. It's a safe way to kind of pick up microorganisms without directly touching them with our hands. This and this kind of go together. This first one is a Bunsen burner, which you may be familiar with from a chemistry course. A Bunsen burner is going to produce a flame, and that flame can be used to sterilize the loop. So we would put the loop in the flame until the loop turns red hot, and then allow the loop to cool before transferring microorganisms. We don't want to contaminate microorganisms when we're working with them. So every time that we put bacteria or another microorganism on this loop, we want to have a way to clean it off. And the most efficient way is to just burn it. We burn it off in this flame. This next one is an infrared incinerator. An infrared incinerator gets really hot using infrared light. The outside metal mesh also warms up, and there can be attachments on here like slide dryers or slide fixers that can be used to warm up slides and help heat fix slides. In the center here, it gets red hot. So we can take a loop, and loops are kind of pictured here as well, and we can stick it in there until it gets red hot, and then we will pull it out and that loop will be considered to be sterile. The main difference between these two is this one has an open flame and this one has the sterilization compartment enclosed. This one is much safer to use in a laboratory or a hospital laboratory, but they both have dangers. So they both get very hot and you have to be careful with them either way. But you don't really want to light up an open fire in a hospital. So we use these more commonly in a clinical lab or a microbiology lab in a hospital. This is various types of media. So this is an agar plate, and an agar plate is used to grow microorganisms on a solid surface. We can look at the shape and size of the colonies, the color of the colonies, and colonies are just small components of bacterial growth or fungal growth. So we can look at the growth on the plate. This is a broth tube. We call it broth. They used to use chicken broth, and today's broth has a whole bunch of different chemicals and nutrients in it, but it's not that different from chicken broth. So this type of broth will allow the microorganisms to grow in liquid medium. This is an incubator, and incubators have all sorts of different shapes and sizes and capabilities. Some incubators use ambient air, meaning that they use the regular air that we breathe in. Other incubators are anaerobic incubators. They use a high concentration of carbon dioxide to grow anaerobic bacteria. But nonetheless, the purpose of the incubator is to hold the temperature steady to allow the microorganisms to grow. Microorganisms, as we'll learn later on, have an optimal growth temperature. So if we hold them at that particular temperature, they'll grow really fast. This is a hood. A hood is used for working with microorganisms that have the potential to aerosolize or spread. So if we want to transfer molds from one medium to the next, we're going to want to put that in a hood because molds have spores, and those spores can aerosolize. They can get in the air, and if you breathe them in and you happen to be immunocompromised, you can get really sick from those. So, or even if you're not immunocompromised and you have allergies to mold spores, you can also have severe allergic reactions. So we like to contain everything in a hood. A hood can also be a fume hood for containing chemicals and so forth. This is a pipette, and a pipette is used for transferring liquids. Pipettes can be set to very specific quantities so that you can transfer specific quantities of liquids. This tip that you see at the end of the pipette is disposable. So usually we use it for a one-time transfer and dispose of the tip. This is a cell spreader. A cell spreader is used to spread cells around on a solid medium. So if you remember that agar plate we had, a cell spreader can be used to push the cells around on that solid medium. This is a microscope, which is the predominant focus of today's, this lecture series. So a microscope is used to visualize microorganisms. This is an autoclave. An autoclave is used to sterilize substances. In a hospital, they may use an autoclave to sterilize surgical tools. In a microbiology lab, we use this to sterilize culture medium. When we use broth media, we want it to be sterile before we start our experiment. When it's done, we also want to sterilize it so that all of the bacteria or fungi that we grew are killed so that they don't have the potential to spread around the lab. So this is kind of like a sterilization chamber. And this one is a microscope slide with a cover slip. So microscope slide and a cover slip are used to observe microscopic organisms in a microscope. So microbial dimensions. The dimensions of macroscopic organisms are given in terms of meters and centimeters. So the average person is a couple meters tall. Dimensions of microscopic organisms are measured from millimeters to micrometers to nanometers. Viruses are 20 to 800 nanometers. The smallest bacterium is 200 nanometers. Yeasts are typically 3 to 4 micrometers, and protozoa are 50 to 100 micrometers. These measurements can be very important, especially if you're working in a medical microbiology lab using in the parasitology sector. Parasites lay eggs, and sometimes their eggs can look very similar. So if we're talking about parasitic worms, or even if we're talking about protozoans that have cysts or trophozoites, their shape can look very similar. And sometimes the only way to properly distinguish them is by measuring their size. So microscopes sometimes have these micrometers that you can measure the size of the microorganism in the field. So knowing these different sizes can be important in that field. For the purposes of this lecture and this course, you don't need to be concerned about the numbers and what nanometer, micrometer, and so forth mean, but just have an idea that, all in all, these are important concepts. So size of things. So a louse can be seen with the naked eye, and a reproductive structure of bread mold can be seen with the naked eye, and they're about 1 millimeter. Giardia lamblia protozoan, so this guy, we'll learn about him later on in this course, is about 100 micrometers. A red blood cell is a little bit smaller. Saccharomyces cerevisiae is a yeast that we use in baking and brewing. It's about 10 micrometers. Then we have E. coli, mycoplasma, and then viruses. HIV is a pretty large virus. We have polio, the average flagellum of a microorganism, a prion, the diameter of DNA, an amino acid, and then a hydrogen atom is 0.1 nanometers. So that just gives you a relative idea of the different sizes of substances. Magnification. So a clear glass sphere can magnify most objects, results from a complex interaction between visible light and waves of the curvature of the lens. So I'm sure you've all used a magnifying glass before. The curvature allows for the magnification of the image. Refraction is defined as bending or changing in the angle of the light ray as it passes through a medium such as a lens. The greater the difference between the two substances, the greater the refraction. So if you've ever stuck a stick in water, the stick looks like it's bending. And that has to do with the way that the light is able to move through the air versus the way that the light is able to move through water. Since it moves through water slower than air, you see a difference in the two images. So the formation of an image occurs when an object is placed at a certain distance from the spherical lens and illuminated with light. Depending on the size and curvature of the lens, the image is enlarged to a certain degree, and it's called the power of magnification, or X or times. The first microscopes just had a single lens, few working parts, Later, they added a second lens, a lamp in the base, and a condenser to focus the light into the specimen. And this is a picture of all of the different parts of a microscope. So we have the ocular lens or the eyepiece, the body, the nose piece holds the objective lenses, and the nose piece allows those objective lenses to toggle to the different magnifications. You have a stage, also known as the mechanical stage, a stage condenser, an aperture or diaphragm control, a base with a light source, and sometimes there's a field diaphragm lever that allows you to adjust the light source, the light intensity control, the coarse adjustment, the fine focus, and then stage adjustment knobs, and you also have the arm. When transporting a microscope, you always want to have one hand on the arm and one hand on the base.